Hello everyone, this is MMBS111, Introductory Microbiology, and we're going to be discussing genetic regulation of protein synthesis. This is mini lecture 8.3. There are 3 learning outcomes for this mini lecture. First, define the term operon. Second, explain at least one advantage an operon provides to bacteria cells. And finally, highlight the main points of lac operon operation. So there are a number of control mechanisms that are used by cells to ensure that genes are active only when the products that they code for are required. So enzymes are produced only when they're needed. Regulation in bacteria, archaea, and eukaryotes involves these 3 mechanisms: antisense RNAs, microRNAs, and riboswitches. We're not going to get into the details of these mechanisms. Instead, we're going to focus on an important mechanism that bacteria use in order to regulate their genes and protein synthesis. So bacteria have something called an operon. They are only found in bacteria. Eukaryotic cells do not possess these. And they consist of a coordinated set of genes that can be regulated as one single unit. So we don't have to regulate each gene individually, but this sequence of genes can be regulated together. They can be inducible or repressible. So if you look at the image there on the right-hand side of the screen, you can see that an inducible operon is turned on when the metabolite is present. A repressible operon is turned off when the metabolite is present. And we're going to talk specifically about the lac operon in just a minute. Catabolic operons are induced by the substrate of the enzyme or the enzymes for which the structural genes code, and they are only produced when the substrate is present. So these would be inducible operons. Repressible operons, as I said, generally code for anabolic enzymes, and they are turned off by the product that is synthesized by the enzyme. Remember, catabolic means to break down. Anabolic means to build up, as we learned in chapter 7. So there is one operon that is found in E. coli that has been studied fairly extensively by scientists, and this is called the lactose operon or the lac operon, and it's one of the best understood mechanisms for explaining this sort of genetic regulation. There are 3 features of the lac operon or any operon. There must always be a regulator, and the regulator is composed of a gene that codes for the protein capable of repressing the operon. And it is found either before or after the operon, so it's found outside that sequence. There is also a promoter that is recognized by RNA polymerase, sorry. This is where RNA polymerase is going to attach and then begin the translation of this section. And then there's an operator, and this is like an on-off switch. So if the repressor is bound, then RNA polymerase can't do its job. The final component of any operon are the genes, and these are the genes that are regulated by the regulator and the operator. And that is the end of section 8.3. So make sure you go through these questions and that you can answer them correctly. If you need to watch the video again, please do so and review the learning objectives. Remember, that's where all of your exam questions are going to come from. Our critical thinking question presents an operon. This is a diagram of the TRP, or tryptophan operon. The question, two questions actually. Based on the diagram to the right, what type of operon is the tryptophan or TRP operon? Is this a inducible operon or a repressible operon? And based on the diagram as well, what do you think regulates the TRP operon? So this is an example of a question you might see on the exam. Make sure that you can go through this and respond appropriately. And hopefully this video was helpful.